Hello everyone, welcome back to the class. In yesterday's class, we learnt about lysosome. It is a single membrane bound organelle which is famously called as suicidal bag because of its property of degradation of components, cellular components, extracellular, intracellular components, and it is heterogeneous in structure and greatly vary in size and shape. It has acidic internal pH of approximately 5 and has hydrolytic enzymes like protease, nuclease, glycosidase, lipase, phospholipase, phosphatase, and sulfates. So the vacuolar H plus ATPase in lysosomal membrane is used to maintain the pH inside the lysosome, that is acidic pH, which pumps the H plus ions. There are two types of lysosomes, that is primary lysosome which do not contain any material for intracellular digestion, and that second one is secondary lysosome which contains material that are undergoing digestion or that have already been digested. So there are two types of digestions happening in the lysosome, that is autophagy and heterophagy. Autophagy means the degradation of cytoplasmic components, whichever the cytoplasmic components which are not necessary for the cell, that degrades with the help of autophagy pathway. Heterophagy is the thing where the external foreign body is first engulfed into the cell and then it is broken down using this lysosome. Heterophagy it is having two different pathway for bringing that extracellular material into the lysosome, that is engulfment. That are two types, phagocytosis and pinocytosis. Phagocytosis is where in phagocytic cells such as macrophages and neutrophils, so they engulf large particle by the process of phagocytosis. During this phagocytosis, first a single membrane phagosome is generated and this compartment fuses directly with the lysosome. This is lysosome and the pink one is phagosome, which is single membrane. So whichever hydrolytic enzymes are present in the lysosome, it directly degrades the component which is present inside the phagosome. So this association or this assemblage is called as phagolysosome. Phagolysosome, due to which the phagocytosis is happening. After this engulfment, after the lysosome has formed, it then it goes inside the lysosome for complete degradation. So next one is pinocytosis, where all eukaryotic cells continually internalize fluid substance in small pinocytic or endocytic vesicles. So the best example for pinocytosis is the late endosome pathway. So whichever protein which has been targeted to the endosome will engulf the vesicle which is containing that protein or a hydrolytic enzymes. So the vesicles first break down. So here the major part which is played by endolysosome. First, how this endolysosome forms? So there is endosome. See here in this picture, there is endosome having hydrolase and intraluminal luminal vesicle inside the late endosome. So the vesicles get fused or the vesicles get degraded, the membrane or that coat gets degraded. Whichever proteins which are present inside that vesicle comes out, and after that endosome will fuses with the lysosome. That lysosome is having hydrolytic enzymes. Of course, in whatever proteins which are present inside the endosome get degraded with the help of lysosome. After that, the digestion of contents happens inside the lysosome. This is the process of pinocytosis we saw yesterday. And there is another one, autophagy. It is the intracellular degradation process of cytoplasmic constituents within lysosome. So how it happens means, first there is the formation of phagophore, which is a single membrane. So then it develops into autophagosome. I'll write it in a diagrammatic way. First there is a phagophore which is of PHP, phagophore. I'll write it as phagophore, or just a minute, I'll write it like this only, phagophore. Then it develops into double membrane bound autophagosome, which is surrounding a portion of cytoplasm. This autophagosome will fuse with endosome. Autophagosome is fusing with endosome. This is endosome, and this association is called to be amphisome. The complete autophagosome or amphisome fuses with the lysosome. This fuses with the lysosome. We can write like this in a small way, endosome, and there is autophagosome. So the lysosome will result in the breakdown of inner membrane of the autophagosome and degrade the material. The resulting macromolecules are released and recycled in the cytosol. This is what the autophagy process happening. So we even saw the diagram, and we saw the flow chart of how it happens. So this is the diagrammatic representation, and these are the images showing that lysosome are of varied size. So some lysosome participate in exocytosis, that removal of the material outside the outside the cell. That the best example is melanocytes, which produces melanin, and it has a specialized lysosome which produces and stores melanin pigment. These pigments containing melanosomes release their pigment into extracellular space of the epidermis by the exocytosis. In this image you can see the clear cut differentiation of endocytic pathway and exocytic pathway. So today we will be learning about vacuoles, the another most important organelle which is exclusively of plants and fungal cells. So vacuoles are found in plants and fungal cells. These are very large fluid filled vesicles. If you remember the plant cell, the vacuole takes the most of the part of the cell. So the complete majority part of the cell is filled with vacuoles. So they are surrounded by a single membrane called tonoplast. So it is also having a membrane which is known as tonoplast. It is similar to lysosome. Similar to lysosome in the sense it is also helping in degradation, and it is evidently having hydrolytic enzymes, protease, phosphatase. The lumen of the vacuole has the acidic pH, which transport ATPase is helping for the maintenance of acidic pH. Remember, it is H plus ATPase, where it transports H plus, or it pumps the H plus inside the cell for maintaining the acidic pH. The plant vacuole contains water, dissolved organic ions, organic acids, sugars, enzymes, secondary metabolites, etc. So this is the image showing the vacuole. See, the whole vacuole is present in this image. So next important organelle is mitochondria. So it is famously called as powerhouse of the cell. So the mitochondria is the term coined by C. Bender. It is a energy converting organelle where it produces energy currency called ATP, which is very much important for plant and animal metabolism. So mitochondria is present in both animals and plant cells. So they produce cellular energy in the form of ATP, and they are the sites of aerobic respiration. They are membrane bound, mobile and plastic organelle. They are double membrane bound structures with outer and inner membrane. So you can see in this image there is outer membrane and where is inner membrane? There is inner membrane also. So the space between the outer membrane and inner membrane, here only. So inner boundary membrane. So that space between outer membrane and inner membrane is known as intermembrane space. And inside the inner boundary membrane there is a matrix. There is a matrix which is a larger internal space which contains double-stranded DNA, mitochondrial ribosomes and various proteins. And there is a cristae here. So what are these cristae? They are internal invaginations of or convoluted folds of membrane which increases the membrane's surface. To write it in a systematic way, first the mitochondria is having the outer membrane, and there is inner membrane also. Inner membrane, so outer membrane is somewhat uniform in nature. There is no invagination, so it is fairly smooth, which protects the organelle and contains specialized transport proteins such as porins. So that porins will allow free passage of various molecules into the intermembrane space. And also this porins or voltage dependent anion selective channels are also there along with this porins, which helps and allows the passage of small molecules across the mitochondrial outer membrane. So there is inner membrane also. Inner membrane is somewhat having foldings here and there. So it is having foldings here and there, and that foldings, internal foldings are known to be cristae. And which greatly increase the membrane surface. They are very impermeable to many solutes due to the presence of high content of phospholipids. So the phospholipid present in the cristae is known as cardiolipin. It is a single word, cardiolipin. Which are the phospholipid present in the mitochondria? That is cardiolipin. There are two faces of this membrane. So imagine, I'll write only inner membrane here. Sorry. So this is the inner membrane, and it is having two face or two side. So this is the N side and P side. So there are two sides of the inner membrane of mitochondria. The middle portion is the matrix side. So as it is matrix, so the inner side is N side, the outer side is P side. Okay. So the inner side is N side, outer side is P side. So surrounding it there is the outer mitochondrial outer mitochondrial membrane, and this is the intermembrane space. Okay, and inner membrane contains enzyme complex, that is ATP synthase. ATP synthase is the most important enzyme complex through which ATP synthesis is happening inside the mitochondria. It is also known as F0 F1 particle. F0 F1 particle, and also there is another name for ATP synthase, that is oxysome. So the major part which is performing, the major function which is performed inside the inner membrane is ATP synthesis. And there is matrix also. The inside matrix is containing a double-stranded DNA, which is particularly circular, and they are having mitochondrial ribosomes. So mitochondria is the organelle which is having DNA of its own, ribosome of its own. So particularly to say, the mitochondria is having ribosome, that is empty ribosome, mitochondrial ribosome, and there is DNA which is empty DNA, empty ribosome, empty proteins. So these are specific for mitochondria. So this is what I explained right now. So this is the image. See, outer membrane, cristae space, intermembrane space, inner boundary membrane. There is cristae involution or convolution of or foldings, cristae junction, the matrix, inner matrix. So mitochondria is a semi-autonomous organelle. Why it is called semi-autonomous organelle is they have their the reasons for calling mitochondria as a semi-autonomous organelle is it is having its own DNA, which is circular. So that is referred as mtDNA. They also possess their own ribosome. So if there is a ribosome, it is evidently said that they synthesize their own protein. So they are not dependent on nucleus or ribosome, cytoplasmic ribosomes. So these organelles are dynamic in the state. They are not static. They don't remain same. So there is frequent changes. So they divide, fuse, change shapes. So mitochondria also multiply. So it divides. So I'll write it here. Divides and changes its shape, or divides, fuses. So and also the fourth point is, so they multiply also. Multiply with the help of binary fission. I'll write it as BF, binary fission. And the evolutionary theory says or suggests that mitochondria has evolved from bacteria, because some of the evidences like similarities with the prokaryotic characters, especially the genetic system, suggest that mitochondria has evolved from bacteria. The theory which suggests that mitochondria has evolved from bacteria is known as endosymbiotic theory, which suggests that mitochondria has evolved from prokaryotes, especially from bacteria. The person who proposed this theory is Lynn Margulis. So endosymbiotic theory suggests that mitochondria has evolved from bacteria due to similarities with the prokaryotic character, especially the genetic system is suggested by Lynn Margulis. So according to this theory, mitochondria supposed have evolved in eukaryotes from endosymbiotic association of purple photosynthetic bacteria. So this theory suggests that mitochondria has evolved in eukaryotes. So mitochondria is present in eukaryotic cells, right? It has been developed, or it has been evolved from endosymbiotic association. Symbiotic association with which type of bacteria? That is purple photosynthetic bacteria. Now Generally tell me that photosynthetic bacteria suggests that bacteria is having chloroplast. Bacteria is having chloroplast, or the photosynthetic apparatus to perform photosynthesis. So we can say that photosynthetic bacteria is having a photosynthetic machinery, and they have evolved along with the other organelle, that is mitochondria. So the captured cell, endosymbiont. So imagine a bacteria anaerobic cell. So imagine a anaerobic cell which is not having the potential to produce its own food. So anaerobic cell derived from an archaean, and there is a primitive nucleus in it, and there is an aerobic bacterium also. Aerobic bacterium, it is having the bacterial outer membrane and bacterial plasma membrane. So it is aerobic bacteria. This is anaerobic cell. Okay, so there is engulfment, or there is capturing of this aerobic bacteria inside this anaerobic cell. So first, aerobic bacteria gets inside the anaerobic cell derived from an archaeon. Okay, after that there is compartmentalization happening. Compartmentalization in the sense, so the aerobic bacteria loses its membrane, outer membrane derived from it derived from the archaeal cell, and it has developed into aerobic. So the anaerobic cell has now developed into aerobic, because the apparatus, or the organelle which is responsible for respiration has been incorporated. So there is internal membrane formation once the engulfment or integration of the aerobic bacteria inside that anaerobic cell. So to explain this image would be enough, I think. See here. An ancestral anaerobic predator cell, an archaeon, is thought to have engulfed the bacterial ancestor of mitochondria, initiating the symbiotic relationship. Clear evidence of a dual bacterial and archaeal inheritance can be discerned today in the genomes of the all eukaryotes. So this is a eukaryotic cell. The eukaryotic cell has been derived from the prokaryotic origin, and the bacterial the anaerobic predator cell is thought to have engulfed the bacterial cell. So this theory was put forth by Lynn Margulis, suggesting that mitochondria is supposed to have evolved in eukaryotes from the endosymbiotic association of purple photosynthetic bacteria. Okay. And the evidences which support this endosymbiotic theory is mitochondria are self-replicating bodies like bacteria and divide in a manner resembling binary fission in bacteria. And also mitochondria are surrounded by two membranes, outer membrane and inner membrane. The innermost of this membrane are similar in composition to bacteria. Mitochondria have their own DNA which is structurally similar to bacterial DNA. Mitochondrial ribosome, enzymes and transport systems are all similar to those bacteria. Mitochondria are of approximately the same size as bacteria. Protein synthesis in mitochondria is inhibited by a variety of antibiotics like chloramphenicol, tetracycline, erythromycin that also inhibit protein synthesis by bacterial ribosome. Here you can see both mitochondria and chloroplast are endosymbiotically formed or evolved organelle. Okay. So mitochondria are inherited cytoplasmically. So in most eukaryotes, mitochondrial inheritance is uniparental. Uniparental in the sense, most precisely maternal inheritance. So the female gametes contribute the mitochondria to zygote. In some eukaryotes mitochondria inheritance is biparental. Both parents contribute mitochondria to zygote. So remember this point, mitochondria are inherited cytoplasmically. Next, another one special organelle is hydrogenosome. Where are these hydrogenosomes present? So in some proteins, protists like Trichomonas vaginalis adapted for anoxic. Anoxic means there is no presence of oxygen, or microaerobic condition. If there is oxygen, so it is in less amount. So that condition in which they lack mitochondria. So these organisms have specialized organelle involved in synthesis of ATP and hydrogen. So therefore that organelle is known as hydrogenosome. Hydrogenosomes are the site of pyruvate fermentation coupled with ATP production via substrate level phosphorylation. Pyruvate is broken down to acetate, carbon dioxide and molecular hydrogen. These are surrounded by soluble membrane and produce ATP. They produce molecular hydrogen through fermentation, lack cytochromes and usually lack DNA also. So see the aerobic mitochondria here move to an anaerobic environment. Other terminal electron acceptors are present here. Anaerobic mitochondria, it becomes anaerobic mitochondria, adapt to an anaerobic environment forming a hydrogenosome where it synthesizes ATP and hydrogen to cope up with that condition. Another one is mitosomes. So what are these mitosomes? These are found in organisms that do not have mitochondria. This organelles is of mitochondrial origin. They are double membrane bound organelle found in some unicellular eukaryotes including entamoeba, histolytica, that is amoeba, and microsporidia. The name mitosome proposed to indicate that the organelle are highly reduced, cryptic, small mitochondria, we can say. So mitosomes are the tiny units of mitochondria. So that's the class today, and in tomorrow's class we'll be learning about targeting of mitochondrial proteins. Thank you.